Dear students, and today we are going to see the questions we can expect from the particular topic, the biomolecules. So as I told you, there are so many segments we have seen: polysaccharides, carbohydrates, nucleic acids, lipids and enzymes. And today we are going to see something about the questions we can expect from the topic, the nucleic acids and the related things. And see the first question. Uridine present only in RNA, that we know. Okay, uridine present only in RNA is a nucleoside, nucleotide, purine and pyrimidine. Okay, you have to choose the correct answer for the term uridine. We will recall the thing. What is nucleoside and nucleotide and purines and pyrimidine? As we all know, in case of nucleic acids, there are two types of nucleic acids we all know, DNA and RNA. Okay, so what is the major difference between these two things? DNA, that is refers to deoxyribonucleic acid, RNA refers to ribonucleic acid. Why we are calling this as deoxyribonucleic acid? Because over here the sugar, the ribose sugar is deoxy sugar, deoxyribose sugar. Over here the normal pentose ribose sugar. Okay, so this is the major difference. So apart from this difference, the one more major difference over here, as I told you, it is made up of the nitrogenous bases. There are adenine, guanine, thymine, cytosine and uracil. These are called nitrogenous bases. We can classify these nitrogenous bases into two major classes, purines and pyrimidines. Okay, so adenine and guanine are purines, thymine and cytosine and uracil are pyrimidines. Okay, so this is what you have to remember. So whenever you are talking about a nitrogenous base, you have to remember the term adenine, guanine, thymine, cytosine and uracil. But over here the term given to you is uridine, not uracil. Okay, so now we will come back to the other two terms, nucleoside and nucleotide. So what is nucleoside? So the nitrogenous base plus a sugar. As I told you, it may be a deoxyribose sugar in case of DNA or it may be a ribose sugar in case of RNA. So the nitrogenous base plus the sugar is called nucleoside. Okay, so then what is nucleotide? Plus a phosphate group. Okay, by means of the phosphodiester bond. So as I told you, a nitrogenous base plus sugar plus a phosphate group. Together, these three together, they will form a structure called nucleotide. So the point you have to remember over here, whenever you are thinking about a term nucleotide, that is along with the particular phosphate group. If the phosphate group is present in the particular structure, that is nucleotide. If there is no phosphate group, only the sugar and the nitrogenous base, that is nucleoside. Okay, now you have to remember the terms. As I told you, whenever you are using the term adenine, that is refers to the nitrogenous base. Okay, you are not supposed to get confused with the terms. When you are using the term adenine, this refers to just the nitrogenous base. When you are talking about a particular nucleoside, adenosine, you have to use the term adenosine. Okay, so this is refers to nitrogenous base plus sugar, that is nucleoside. Okay, then what about the nucleotide? Over here you have to use the term adenosine triphosphate or adenylate acid. Okay, so these two terms refers to nucleotide. So the point you have to remember over here, there are different terms we are going to use, slightly different terms we are going to use for nitrogenous base, nucleoside and nucleotide. Okay, so the two things you have to remember over here, nitrogenous base refers to just the nitrogenous base, adenine, guanine, thymine, cytosine and uracil. Nucleoside refers to nitrogenous base along with the particular ribose sugar or the deoxyribose sugar. Over here you are going to use the term end with the particular INE. Okay, so adenosine, uridine, Okay, thymidine, cytidine refers to the particular nucleoside. Nucleotide refers to, over here you have to use adenylate acid or adenosine triphosphate or the uridine triphosphate, like that you are gonna use. So the point you have to remember, now you can choose the answer. Uridine present only in RNA is uridine is not a purine or pyrimidine. As we all know, purine and pyrimidine are just a nitrogenous bases. So you cannot choose this as a answer. Okay, it must be nucleoside or nucleotide. I told you, whenever you are thinking about a nucleotide, it should be come along with a particular phosphate group. So there are no phosphate group. This is not a uridine triphosphate, just a word uridine given to you. So this is not an answer. Uridine in RNA is a nucleoside is the answer. This is how you have to choose the answer for the particular type of question. And see the next question. DNA is composed of repeating units of, over here there are 4 choices given to you: ribonucleotides, deoxyribonucleotides, ribonucleoside and deoxyribonucleoside. Okay, so the differences over here: deoxyribonucleosides, okay, and deoxyribonucleotides, ribonucleoside and ribonucleotides. Okay, so as I explained in the last question, you have to remember DNA refers to deoxy. Okay, so DNA refers to deoxyribonucleic acid. Okay, deoxyribonucleic acid. Okay, so the point you have to remember over here, the major difference between these two, the presence or absence of the deoxy sugar. So the answer must be one among these two, deoxyribonucleotide or deoxyribonucleoside. We can easily eliminate the ribonucleotides. So ribonucleotide and ribonucleosides present only in RNA, not in the DNA. So the choices remain for you: deoxyribonucleotides and deoxyribonucleosides. So I told you, so DNA is a polymer. We all know DNA is a polymer. So polymer of nucleotides, so we can call this as polynucleotides. So what do you mean by polynucleotides? It is made up of the monomers of nucleotides. So there are so many nucleotides, it is being combined with each other by means of the phosphodiester bond, and they are going to form the particular structure called DNA. So the point you have to remember over here, the DNA is made up of deoxyribonucleotide. Okay, not side. Side always refers to without the phosphate bond or phosphate group. So whenever you are talking about a nucleic acid, whenever you are talking about a nucleotides, you have to choose the deoxyribonucleotides. So I'll explain you one thing. So we all know, we are calling this as ribonucleic acid and deoxyribonucleic acid. But I told you the major group present in a particular nucleic acid is a nitrogenous base. But why we are calling this as acid? Okay, so we are calling this as the major group over here is adenine, guanine, thymine, cytosine and uracil. Okay, this is the bulky group, the major group present in a particular DNA and RNA. But we are calling this DNA and RNA as a acid, deoxyribonucleic acid and ribonucleic acid. Why we are calling this as acid, not as a base? Okay, so the point you have to remember over here is the presence of the particular phosphate group. So in a DNA or RNA structure, okay, so the nitrogenous base is being attached with a sugar and sugar is being attached with a phosphate group. Okay, so the external surface is being covered with the phosphate group. That phosphate group can be ionized to form a acid structure, so that the nature of the particular DNA and RNA in a liquid environment, in a cytological environment, a body fluid environment, it is in the form of the acid. So that only we are calling this as deoxyribonucleic acid and ribonucleic acid. So you have to remember the thing, whenever you are seeing the term acid, you have to remember the presence of the phosphate group. Whenever you are thinking about a phosphate group, you have to remember the term nucleotide. Okay, so from this explanation you can easily select the particular answer. The question over here, the DNA. What is the DNA full form is deoxyribonucleic acid. So acid is present over here. So whenever you are thinking about acid, you have to remember the term, the presence of the particular phosphate group. So whenever the phosphate group is present, you have to recall the term nucleotide. So this is D. D refers to deoxy. So here you can easily select the answer deoxyribonucleotides for the particular question. And see the next question. ATP is a, okay, so choices are nucleoside, nucleotide, nucleic acid and vitamin. Okay, so among the choices we can easily eliminate the vitamin. ATP is not a vitamin. We all know ATP is a energy carrier molecule. It is going to provide the energy throughout the cell, and of course I told you ATP is a part of the particular nucleic acid. It is present in the DNA and RNA is in the form of, as I told you, A refers to adenine. Okay, so what is the full form of ATP? Adenosine triphosphate. Adenosine triphosphate. Okay, so it is present in both DNA and RNA as a adenosine triphosphate. So the point you have to remember over here, ATP is a nucleoside, nucleotide, nucleic acid and vitamin. So I told you, whenever you are talking about a nucleic acid, it is talking about a polymeric structure of the particular nucleotides. Okay, so ATP is a single unit, so you cannot use the term nucleic acid for the particular single unit. Okay, so you have to choose the answer from these two choices, nucleotide and nucleoside. I told you the full form for the ATP is adenosine triphosphate. Whenever you are seeing the term phosphate, you have to recall phosphate is always associated with a particular nucleotide. Whenever you are seeing the term phosphate, then you can easily combine or you can easily connect the particular term with the nucleotide. So ATP is a nucleotide, not a nucleoside. Just the term adenosine is nucleoside. Adenosine triphosphate is a nucleotide. Okay. Now see the next question. Adenine is a purine, nucleoside, nucleotide and a pyrimidine. Okay. So this is a straightforward question but a bit confusing question. So adenine, we all know adenine is a kind of nitrogenous base present in a particular DNA and RNA. And you have to choose, there are no choice over here as nitrogenous base. You have to choose from the purine, nucleoside, nucleotide and pyrimidine. Okay, and we'll start from this nucleoside and nucleotide. We all know it is a combination of nitrogenous base plus sugar plus, if the phosphate is present, it is nucleotide. If there is no phosphate, it is nucleoside. Okay, we all know there are two groups of nitrogenous bases, purines and pyrimidines. And what are the bases present in case of purines? Adenine and guanine. In case of pyrimidine, thymine, cytosine and uracil. Okay. So if the term is adenosine, you can choose the answer nucleoside. If the term is adenosine triphosphate or adenylate acid, you can choose the answer nucleotide. Now you can eliminate these two because the given term for you is adenine. Okay. So you have to choose whether it is a purine or pyrimidine. We all know adenine belongs to purine along with the guanine. Adenine and guanines are purines. Thymine, cytosine and uracil are pyrimidines. So the answer is purine. Okay. See the next question. RNA does not possess. Okay. So this is also a straightforward question for you. Uracil, thymine, adenine and cytosine. Okay. So I told you, again I will repeat to you, the major difference between the DNA, the presence or absence of the deoxyribosugar or the ribosugar. So apart from the first major difference, the second major difference over here, in case of DNA, the nitrogenous bases are adenine, guanine, thymine and cytosine. In case of RNA, adenine, guanine, cytosine and uracil. Okay, so you can easily see the differences over here. The purines are similar for both the DNA and RNA. So in both DNA and RNA, the purines are adenine and guanine. But in case of pyrimidines, over here in DNA, thymine and cytosine. In case of RNA, there are cytosine and uracil. So over here, the thymine is being replaced by the uracil. Okay. So uracil is only present in RNA. That is the point you have to remember. That is the thing you have to remember over here. So RNA does not possess, uracil is there, adenine is there, cytosine is there. Okay. So the answer is thymine. So thymine is absent from the particular RNA structure, ribonucleic acid. So this is how you can expect the question from the particular nucleic acid, and we are going to continue the patterns of question we can expect from the particular topic, the nucleic acids. See the next question. So in a double helix of DNA, the two DNA strands are, so over here they are talking about the double helical nature and the coiling nature of the particular DNA structure. Okay, so see the choices: coiled around a common axis, coiled around each other, coiled over a protein sheath, and coiled differently. Okay, so we all know DNA is made up of two strands. Okay, two strands of polynucleotides. What is polynucleotides? It is made up of the monomers of nucleotides. So what is polynucleotide? Made up of so many nucleotides as a chain. Okay, what is two strands? There are two strands of the particular polynucleotide. Okay, so see the choices over here. So in three choices you have the term coiled. Okay, you have to select the choice from here. Of course, all the four choices they are possess the term coiled. Okay, so over here, whenever you are talking about a DNA, the structure of DNA itself, you are not supposed to confuse with the protein. Okay, so the chromosome, chromatin, the nucleosome. Okay, so whenever you are using the term chromosome, chromatins, nucleosomes, you have to remember the DNA is coiled around the particular protein compound, histones. Okay, but when you are talking about the structure of DNA alone, you are not supposed to confuse with the histone. Here there are no proteins involved in the particular DNA structure. Okay, so the DNA structure is being proposed by the Watson and Crick. Okay, so according to this structure, there are no proteins involved for the particular DNA. Okay, so just you have to remember the DNA is made up of nucleotides. That particular nucleotides present as a polynucleotide strands. There are two polynucleotide strands. They are being coiled around each other. This is what you have to remember. So you can easily eliminate the fourth choice. Coiled around or coiled over the protein sheath is the wrong answer. Okay, so this is correct only in case of nucleosomes, chromatin and chromosomes. Over here we are talking about only the DNA structure. See the question again. In double helix of DNA, this is over here they are talking about only the DNA structure. Okay, so just eliminate the protein sheath covering. So see the remaining three choices: coiled differently, coiled around a common axis, coiled around each other. So all the three choices are seems correct over here. Okay, coiled around each other and coiled around common axis, coiled differently are same. But over here they are not identical. They are similar terms and you have to choose the correct among these three. So coiled around a common axis means there is a central common axis. Okay, around the common axis that particular DNA strands are running around each other. This is what you have to remember. Okay, around the common axis they are running around each other. Coiled differently means that is some other term just opposite to the particular common axis. Coiled around each other is correct. When it is correct, when there is no coiled around the common axis, then you can choose the coiled around each other. But we have a choice over here, coiled around the common axis. Okay, then you can eliminate these two choices. So over here you have to remember in DNA there are two strands. Those two strands coiled around each other. Up to this is correct. Coiled around each other in a common access, then you have to choose the choice the A. Okay, and see the next question, which is not consistent with the double helical structure of DNA. Okay, so over here there are 4 choices given to you and you have to choose which one is inconsistent with the particular DNA helical nature or helical structure. So the choice 1, this is not a hydrogen bond, we have mentioned over here, this is equal to, adenine is equal to thymine, cytosine is equal to particular thymine. Okay, come back to this. Density of DNA decreases on heating, and see the next choice, adenine plus thymine, cytosine plus guanine is not constant, both A and B. Okay, so in so many questions you can you can see the choices like this. You have to choose 2 or 3 choices at the particular time for the particular question, and see one by one. When here we are talking about the double helical structure or double helical nature of the particular DNA. Okay, so in a DNA you have to remember there are 4 bases present. I told you it is made up of adenine, guanine, thymine and cytosine in a particular composition for the particular sequence. Okay, and we all know adenine always pairs with thymine, guanine always pairs with cytosine. Adenine always pairs with thymine, guanine always pairs with cytosine. Over here we are mentioning this as hydrogen bonds, not as equal to. Okay, adenine pairs with thymine by means of 2 hydrogen bonds, guanine pairs with cytosine by means of 3 hydrogen bonds. Okay, so remember this and come back to this. Adenine equal to thymine, cytosine equal to thymine. This is not a correct thing. Cytosine is equal to guanine, not a thymine. So this is a wrong choice. Adenine is equal to thymine is correct, because as I told you, adenine always equal to, adenine is always pairs with thymine. So adenine and the number of adenine is always equal to thymine. As I told you, you have to remember this thing. This is a double helical nature, double stranded structure. Okay, so in a particular one strand, if the adenine is present, the opposite strand must possess the particular thymine. Then only it will make a hydrogen bond over here. So while you are counting the number of adenine, it is always equal to number of thymine. Okay, so the number of adenine is always equal to thymine, number of guanine is always equal to cytosine. This is what you have to remember. So over here adenine is equal to thymine, up to this correct, but cytosine is not supposed to be always equal to particular thymine. Cytosine is always equal to guanine. So this is a wrong choice for the particular question. So over here density of DNA decreases on heating. So what is density? So I told you it is a double helical structure and it is being filled with an nitrogenous bases connected by hydrogen bond. So what will happen while you are heating the particular DNA in a solution? That heating of DNA will denatures the particular structure. Why it is denatures the particular DNA structures? It is going to break the hydrogen bonds. It is going to break the hydrogen bonds in between the particular nitrogenous bases. So if there are no hydrogen bonds, it will release the 2 strands apart. So it will increase the density of the DNA. So this is a wrong choice. Okay. So both A and B are wrong. So you can eliminate the particular choice. So answer is A plus T and C plus G is not constant. That is the correct answer. And we are going to explain the particular thing in the next question. A plus T and C plus G is not constant. Okay. Come back to the next question. I told you. Which of the following nucleotide sequence contains 4 pyrimidine bases? Okay. So this is a very straightforward question. So over here you have a DNA sequence like this. Okay, they have given some sequences. I told you we can write the DNA sequence by means of their alphabets. I told you there are 4 bases: adenine, guanine, thymine and cytosine. Suppose you want to write a DNA as a sequence, you have to start from a 5' end to 3' end. Okay, then you can write the sequence like this. You can write the sequence like this. Okay, so 5' is refers to a particular end, and from the 5' you are proceeding with the direction to the 3 prime, you have written the particular DNA in a sequence in a particular paper, a written form of the particular sequence. So over here there are 4 sequences given to you, there are 3 sequences and a choice given to you, and you have to choose a sequence which contains 4 pyrimidine bases. I told you there are 2 types of bases, purines and pyrimidines. What and all purines? Adenine and guanine. Thymine, cytosine and uracil are pyrimidines. Okay. So with this knowledge you have to count the pyrimidine bases in the particular sequence. So over here, so what are the pyrimidines? Thymine, cytosine and uracil. Thymine, cytosine, thymine, cytosine. Okay, over here cytosine, cytosine. Only 2 pyrimidines are present, so this is not a choice. So over here uracil, cytosine, uracil, only 3 pyrimidines are present, so this is not a choice. So we have eliminated B and C, so B and C of course. So over here you can count 1, 2, 3 and 4 pyrimidines. There are thymine, cytosine, so the correct choice over here A. Okay, this sequence contains 4 pyrimidines. Okay, see the next question. So here we are going to explain the previous question. A segment of DNA has 120 adenine and 120 cytosine bases. Okay, so the question is the total number of nucleotides in a particular sequence. So as I told you, so here you have to remember the term Charkov rule. Charkov rule. So according to Charkov rule, as I told you, adenine, number of adenine is always equal to thymine. The number of guanine is always equal to cytosine. Okay, why this happens? As I told you, adenine always pairs with thymine, guanine always pairs with cytosine. So that the number of adenine is always equal. Okay, you have to remember the term equal. Always equal to thymine, guanine always equal to cytosine. Okay, so I told you in the previous question, the A plus T and C plus G is not constant. So why we are calling this as not constant? I told you. So from this statement you have to remember one thing very clearly. The number of adenine may not be equal to guanine. The number of thymine may not be equal to particular cytosine. Why? Because they are not gonna make bond, hydrogen bond with each other. Okay, so the number of adenine may be differs from the number of guanine. The number of thymine may be differs from the number of cytosine. So that is what we have explained over here. The A plus T content and C plus G content is not constant for the particular species. Of course, we can identify the species based on a particular ratio. Okay, for each and every species, for example, for each and every species, the A plus T and G plus C, the ratio between these two, the ratio between the adenine, thymine and guanine, cytosine ratio is constant for a particular species. For each and every type of species, this ratio is constant. That means the number of adenine and the number of thymine is particular. The number of adenine and thymine is constant for the particular species. In the same species, the number of guanine and cytosine is constant in the particular same species. Okay, so this is what explained over here. The ATCG is not constant. Okay, that is what you have to remember. That means the number of adenine is not equal to cytosine. The number of thymine is not equal to guanine. This is what you have to remember. Come back to the final question. As I told you, a segment of DNA has 120 adenine and 120 cytosine, and you have to find out the total number of nucleotides over here. So from this you can easily predict. So number of adenine over here is 120. So what may be the number of thymine? As I told you, the number of thymine is equal to adenine. Adenine always pairs with thymine. So the number of thymine is of course 120. Why? The 120 adenine base is gonna make bond with 120 thymine. So another hint over here, the number of cytosine is of course 120 over here. Cytosine always first with guanine. So the number of cytosine over here 120. So the guanine must be 120. And the question is the total number of nucleotides in the particular sequence. You are gonna add up all the things. The sum of this 120 plus 120 plus 120 plus 120, 480. Okay. So 120 is wrong answer. 240, 60. The answer is 480. Okay. So this is how you have to select the answer for the questions you can expect from the particular topic, the nucleic acids. So in the next classes we are going to see the question from the some other topics from the particular biomolecules. Hi students, and today we are going to continue the questions from the same unit, the biomolecules, and today we are going to see the questions we can expect from the topic, the amino acids, proteins and enzymes. Okay, so see the first question. Amino acids are mostly synthesized from, okay, just compare the question with the second question. Amino acids are produced from, okay. So for your kind information, both the questions asked from a same year of 1992, I think so, NEET 1992. So you can easily distinguish these two questions. Amino acids are mostly synthesized from and amino acids are produced from. You have to differentiate these questions. I'll explain. Okay, so what are amino acids? As I told you, amino acids are the building blocks of proteins. Okay, so that means the proteins are synthesized from the particular amino acids. So proteins are made up of amino acids. Proteins are made up of amino acids. That means amino acids are building blocks of proteins. Okay, so amino acids, they are going to link with each other by means of the bond, peptide bond. Okay, and they will form the polymeric structure that is called the polypeptide. And the arrangement of the polypeptide chain or a polypeptide chain is called a protein sequence. Okay, so the point you have to remember whenever you are thinking about amino acid, you have to remember the macromolecule protein. So proteins are made up of amino acids. Proteins are made up of the building blocks of the amino acids. So the question over here, the amino acids are synthesized from. That means I told you. So while we are studying the metabolism, I told you there are two reactions, synthesis and the digestion. Okay, so catabolism and anabolism. So over here it is talking about the synthesis. That means the synthesis of amino acids. So as I told you, for the synthesis of proteins you need amino acids. So I told you. So for the protein synthesis there are so many molecules involved. As I told you, it starts from the DNA. It carries the information for the synthesis of the DNA, and the information is being copied as RNA. There are three types of RNA, mRNA, tRNA and rRNA. So the mRNA carries the message for the synthesis of proteins. The tRNA transfers the molecules required for the protein synthesis. That molecules are amino acids. tRNA transfers the amino acids to the protein synthetic site, and rRNA will form the ribosomes. And the point you have to remember over here, from where the particular tRNA transfers the amino acid to the protein synthetic site. Okay, so imagine a cell. Okay, so the protein synthetic site, it may be the rough endoplasmic reticulum, or it may be in the cytoplasm in the free ribosomes. So the point you have to remember over here, the protein synthesis happens at the site ribosomes. Okay, so if the ribosomes present in the cytoplasm, it happens in the free ribosomes. If the ribosome present in endoplasmic reticulum, the protein synthesis happens at the particular rough endoplasmic reticulum. Okay, so the point you have to remember over here, for the synthesis of proteins, you have to transfer the amino acids from one place to another place. From which to which? Normally the amino acids are being synthesized all over the particular cytoplasm, or it is being digested from the food material ingested by the particular cell. Okay, so that kind of synthetic or synthesized amino acid is being transferred to the particular ribosomes to synthesize the particular protein molecule. So what is the question over here? It is talking about the synthesis of particular amino acid. Okay, from where it is being synthesized? What is the precursor molecule for the particular amino acid synthesis? And see the choices over here. Okay, so volatile acids, it is nowhere related to the particular amino acid synthesis. Fatty acid, as I told you, amino acid, they contain the compound called amino group, carboxylic acid group, and there are functional group along with the hydrogen atom at one side. Okay, so that structure does not fit with the particular fatty acid. Okay, so there are two other choices for you, mineral salts. Okay, there are no relationship for the salt with the amino acid. So the answer for you over here, alpha ketoglutarate acid. So I will explain something more for you for this question. So we all know. So what is alpha ketoglutarate acid, from where it is being synthesized? So in the metabolic pathway, okay, in the metabolic pathway, for example from the pyruvate, it enters into the Krebs cycle or TCA cycle. From there it will synthesize alpha ketoglutarate acid, and the particular alpha ketoglutarate acid is being act as a precursor molecule for the synthesis of amino acids. Okay, so whenever you are talking about a synthesis, you are gonna synthesize the amino acid, you have to remember the term alpha ketoglutarate acid. Okay, so come back to the second question. Amino acids are produced from. So this is somewhat confusing part of the question, the term produced. So as an animal being, we are ingesting so many different kinds of food molecules. Okay, for example, we are ingesting carbohydrates, we are consuming oils as lipids. Okay, we are consuming, of course, so many proteins. Okay, so what is the need for protein over here? From protein we are gonna get some molecules required by the particular animal cells. Okay, so what happens over here? The proteins, so whatever macromolecule ingested by us in the form of food, whatever macromolecule ingested by us in the form of food will be enters into the digestion. So what will happen over here? The digestive enzymes gonna act over here, and it will digest each and every type of the polymeric structure. If it is a carbohydrate, what will happen? Polysaccharide will be digested into the monosugars. That monosugar, for example, the glucose will enter into the glycolysis, and from the glycolysis it will synthesize or it will produce into, for example, you have started off with the carbohydrate polysaccharide. Okay, it will enter into the digestion. From the digestion you will get the monomeric sugars, for example glucose, and the glucose will enter into the cycle glycolysis, and from the glycolysis we will derive the energy in the form of oxidation reduction reaction. So from the glycolysis it will produce pyruvate. From the pyruvate it will enter into the Krebs cycle, as I told you. From the Krebs cycle it will enter into the electron transport chain. From the electron transport chain we will get the energy in the form of ATP molecule. This is how our digestive system works. Okay, so we are ingesting some food molecules in the form of carbohydrates, proteins, lipids, and some other macromolecules. From that we are getting the energy as a final product. Okay, so the point you have to remember over here, our question is amino acids are produced from. For example, we are ingesting a protein as a food. We are consuming egg or cereals. What will happen? That protein will be digested into smaller molecules, monomers of the particular protein. So as I told you in a previous question, the proteins are a polymeric structure of amino acids. Okay, so the proteins are made up of building blocks of amino acids. That means polymers of the amino acids will make up the structure called the protein. So if you digest the particular protein, what you will get? You will get the amino acids. That is what you have to remember. Okay, so there are two types of amino acids, or there are two classes of amino acids: essential amino acids and non-essential amino acids. So non-essential amino acids means the body itself can produce the particular amino acid. Essential amino acids means the body has to consume the particular amino acids in the form of food. Okay, so over here your question is amino acids are produced from. Okay, so you cannot produce amino acids from fatty acid, you cannot produce from alpha keto acid. Don't confuse with the term alpha keto acid. Over here the term alpha ketoglutarate acid. Okay, so alpha keto acid refers to any acid which contains the particular keto group. Okay, so this is not answer. You are not supposed to get the particular amino acid from the essential oils, because essential oils belongs to the lipids or fatty acid group. So the answer over here, you have to choose the particular proteins. Okay, so now you can differentiate these two questions. As I told you, amino acid synthesis is from alpha ketoglutarate acid. If you are gonna degrade the particular proteins, you will produce amino acids. Okay, so this is the difference you have to remember in case of the amino acid synthesis and production. Okay, so see the third question, the enzymes having slightly different molecular structure but performing identical activity. Okay, so we have seen something about the enzymes in the introduction classes. So now we will recall something about the enzymes. So there are so many different terms you have to remember about the particular enzymes: holoenzyme, apoenzyme, coenzyme and isoenzyme. So what is holoenzyme? So as I told you in the introduction classes, enzymes are normally made up of so many subunits. Okay, enzymes, as I told you, most of the enzymes are proteins. Just remember, not all. There are some nucleic acids, they can be act as a enzyme, but 99% of, apart from the particular ribonuclease, okay, apart from the particular, as I told you, the ribozymes. So apart from these ribozymes, most of the enzymes are made up of proteins. That means they are made up of the particular amino acids polymer, polynucleotides, polypeptides. So the point you have to remember over here, there are so many polypeptides will make up the particular functional enzyme. So each polypeptide is called as a subunit of the particular enzyme. Okay, so there are so many subunits will make up the particular enzyme structure called holoenzyme. So there are some other term for the holoenzyme is conjugate enzyme. So in other words, we can explain the term holoenzyme. For a particular enzyme, that enzyme will require some other group or some other molecule is being attached to the particular enzyme, then only it will act as a functional enzyme. Okay, that means we can divide the particular holoenzyme into two groups or two sections. Holoenzyme, one is simple protein part. That means that is made up of only amino acids. Plus there are some other non-protein part may be present with the particular enzyme. Okay, plus some non-protein part. Okay, together we are calling this structure as holoenzyme. This simple protein part or made up of amino acid part is called as apoenzyme. Okay, so what is apoenzyme? In a holoenzyme, the protein only part is called the apoenzyme. Okay, so just remember, apoenzymes cannot act as a functional or fully functional enzyme. It needs the particular non-protein part to be attached with the particular apoenzyme, then only it can act as a particular functional enzyme. Okay, so what is holoenzyme? Holoenzyme is a multi-subunit complex. Okay, there are two regions in the particular holoenzyme, protein part and non-protein part. The non-protein part may be organic and or inorganic. Okay, so the point you have to remember over here, apoenzyme refers to the protein part of the particular enzyme. Non-protein part may be organic or inorganic. So we are calling this non-protein part as a cofactors. So already we have seen there are three classes of cofactors in our previous classes. Okay, coenzymes and metal ions and prosthetic groups. Okay, so what are the three groups? Coenzymes, prosthetic groups and mineral ions. We will call this as metal ions. So you can see it over here. Coenzymes are organic part, prosthetic group is a non-protein part, and metal ions are just elements. They are going to act as a coordination. They are going to make the coordination bonds with a particular substrate and product along with the enzyme. Okay, and these three groups are belongs to the category cofactors or the non-protein part. It is being attached to the particular apoenzyme to make the holoenzyme. Now come back to the question. You have a question, enzymes having slightly different molecular structure but performing identical activity. So what and all we have seen? Holoenzyme. There is no relationship with the explanation given in your question. Okay, we can eliminate the holoenzyme. Apoenzyme is related with the holoenzyme. Coenzyme related to the holoenzyme. So we can eliminate the three. Then the choice will be the isoenzyme. So what is your question? Enzymes having the same or enzymes having slightly different molecular structure, identical activity. For example, in a human cell, there are enzymes called lactose dehydrogenase. There are so many different variants of the particular dehydrogenases present. At least 20 to 40 different types or the variants of the particular lactose dehydrogenase is present in a particular single human cell. Okay, so what are variants? They are made up of different amino acid sequence. Different amino acid sequence. So as I told you in the introduction classes, you have to remember the protein structure is based on the amino acid sequence. Okay, so each and every type of each and individual amino acid they have in the particular polar or non-polar property, and based on that property only they will attain a position in a particular protein chain, and based on the position only the protein chain will get the particular structure, and based on the structure only they are going to act as a particular functional molecule. Okay, so over here you are having the different amino acid sequenced proteins. Okay, so that as I told you they are having slightly different molecular structure. Okay, over here what you have to remember? Different amino acid sequence, different structure, but they are having identical activity. All the particular structures they are having the same function, identical. Okay, not similar. You have to remember the term identical. They are having the particular identical functions over here. Okay, and these group of enzymes are called isoenzymes. Iso means same. Okay, so isoenzymes. So what is isoenzyme? You have to remember the isoenzyme. They are the group of enzymes. They are having different amino acid sequences, different molecular structure, but they possess the particular identical functions or identical activity. So see the next question. The enzyme enterokinase helps in the conversion of something. Okay, and see the choices. So whenever you are seeing the term enzyme and a kinase, they are gonna involved in something conversion. Okay, so there are four choices given for you, four conversions. See the conversion over here. Conversion of proteins into polypeptides. That means you are going to digest the protein molecules and you are going to break the particular peptides. Okay, peptide bond. So what happens over here? You are going to digest the particular peptide bond so that you will get the individual peptide units. Okay, so this is what given over here. Proteins into polypeptides. Digestion of protein. The large proteins into individual subunits or individual units of the polypeptides. And see the next choice. Pepsinogen into pepsin, trypsinogen into trypsin, casinogen into casein. The thing you have to remember over here, from the inactive form to the active form, pepsinogen into pepsin, casinogen into casein, trypsinogen into trypsin. Okay, so all are related with one thing, one common thing, proteases. Okay, so what are proteases? The enzymes which can digest or which can act on a enzyme and which can act on proteins. Okay, proteases can act on proteins and it will produce product from the particular protein. Okay, so casinogen is an inactive form of casein. It will be converted into casein. Pepsinogen will be converted into pepsin. But the question over here is enterokinase. Okay, so the another term for the particular enterokinase is enteropeptidase. So the enterokinase and enteropeptidase is always related with the trypsin. Okay, so they are involved in the conversion of trypsinogen into trypsin. So the choice over here, the second one, the trypsinogen into trypsin. Okay, so these are the questions you can expect from the particular amino acid segment and the enzymes. And now we are going to continue with the same segment. The questions you can expect from the same topic, the amino acids and the enzymes and the proteins. And see the next question. The role of enzyme in a reaction is, okay, there are four choices for you: decrease activation energy, increase activation energy, as an inorganic catalyst, none of the above. Okay, and for this question you have to remember, you have to recall the enzyme function and how the particular enzyme works. Okay, so as I told you, what is the role of enzyme? Enzyme act as a catalyst. Okay, so by this term you cannot choose this as an answer, inorganic catalyst, just because the catalyst, the word is present over here, because enzymes are organic molecules. Okay, enzymes not a inorganic molecule, enzymes are organic molecules, so you are not supposed to select the particular in C. Okay, so automatically you can eliminate the answer D. Okay, enzymes are organic molecules, they are catalyst, they can act as a catalyst, or they are catalyzing the reaction from the substrate to the product. So you are having the substrate, you are going to synthesize the product with the help of enzyme. Okay, so what happens over here, as we have seen in our last class? So from an enzyme, so towards the, so over here we are having the energy during the rate of reaction or the progress. So from the substrate you are going to produce the product. Okay, so I told you there are two levels of energy you have to remember: stable and unstable. Stable and unstable. Just remember one thing, both substrate and products are in a stable form. Okay, both substrates and product are in a stable form. But during the transformation, during the biotransformation from the substrate to the product, it will form the unstable complex or unstable form, that is called the enzyme substrate complex or the enzyme product complex. Okay, so both the complexes are unstable. Okay, so what happens over here? First, how the process will happen, what are the steps involved over here? The enzymes go and binds with the substrate, or the substrate diffuses into the enzyme molecule. It will form a enzyme substrate complex. Okay, and the enzymes will changes the making or breaking the bonds. That is what called breaking or making of bonds. Okay, so that is what we are calling as the enzymatic action. So what enzymes are doing over here? The enzymes will change the substrate into the product. How it is changing? Initially it will form the enzyme substrate complex, then it will changes the bonding nature of the particular substrate. It will make or eliminate or break the particular bond, so that it will form a completely different structure from the substrate. That substrate, that product, that is called as a product. That different structure, formed structure is called product. Okay, so that is how the particular enzyme works. So the point you have to remember over here at the energy levels. So over here the energy is at very low. Over here the energy is at very low. So that only we are calling this as stable form. Whenever you are having whenever the particular molecular structure having a higher energy, that is unstable. That is what you have to remember. Whenever the particular molecule in its lowest energy possible state, that is called stable form. Okay, so substrate and product is in a stable form, and it has to transfer the particular unstable form. Okay, I told you unstable means it has higher energy. Okay, so this higher energy state, to attain the particular higher energy state, that particular substrate needs some energy. That energy we are calling it over here the transition energy. Okay, so we are calling it as the term transition energy. To attain a particular transition energy, it needs some activation, so that we are calling this as activation energy. Okay, so this is what you have to remember. We are talking about the activation energy. Okay, so if the substrate is in need of higher activation energy, it will take so much of time to convert it into the product. Okay, so what enzyme has to do over here? The enzyme has to reduce the amount of energy to activate the particular transition state. Okay, so what is transition? Suppose you are entering into a makeup room. Okay, suppose you are as a human being entering into a makeup room. So the makeup room is called as a transition period. So within a makeup period or makeup room, you are getting some makeup, and after the makeup completion, you are gonna come out as a better personality or better looking man. Okay. So what happens over here? The transformation happens within the particular makeup room. Okay. So while you are entering as a substrate and coming out as a makeup or the better personality product. Okay. So the point you have to remember over here, what happens during the makeup period or within a makeup room. Okay. You cannot be sitting within a makeup room for some days or some hours. Okay. You have to come out of the particular room within a whatever possible time, whatever minimum period of time. Okay. So that is what happens during the enzymatic action. The enzymatic catalysis, what it tries to do, it will try to increases the, or it will tries to decrease the particular activation energy, so that it will reduces the time in the particular transition period, so that the product formation will be faster than the non-catalytic reaction. So come back to the choices. Decrease activation energy, increase activation energy. Okay, there are two choices for you. Okay, so the choice will be the decrease activation energy. Why? So that only it can decrease the transition period, so that only it can decrease the time in the particular transition period, particular unstable period, so that it can speed up the reaction from the substrate to the product. So the choice over here, it will decrease the activation energy. So see the next question. Some enzymes which are associated in converting fats into carbohydrates. We should have some knowledge about the particular conversion of fat. Conversion of fats into carbohydrate. So just remember one thing. From the normal pathway, in case of animal being, we are normally consuming the food in the form of carbohydrate, that is polysaccharides, for example rice. Okay. So from the polysaccharide we are going to synthesize the, as I told you, the monosugars, for example glucose. From glucose we are entering into the glycolysis metabolic pathway, then pyruvate, Krebs cycle, electron transport chain, then energy. Okay. So this is what the normal steps for the energy synthesis for any animal cell. Okay. From the consumed carbohydrate polysaccharide, it will synthesize the monosaccharides. From the monosaccharide glycolysis, from the glycolysis Krebs cycle or TCA, then electron transport chain, then energy synthesis. So the question over here, the synthesis of carbohydrates from fat. Okay. So when there are no enough carbohydrates available in a particular animal body, so what will happen? It will try to synthesize the particular carbohydrate from the available fat. Okay. So that is what happened over here. So the straight question over here. Okay. What is the exact location of the particular conversion? Fats into carbohydrate. Okay. So there are four choices over here. Golgi bodies, liposomes, microsomes and glyoxysomes. Okay. Golgi bodies, as we all know, as we have seen in the last chapter, Golgi bodies are involved in the modification of lipids and proteins, not in a synthesis of carbohydrates. Okay, so you can eliminate that. Microsomes and liposomes are nowhere related to the synthesis of the particular carbohydrate. So the choices over here, the automatic choice over here is glyoxysomes. Glyoxysomes, as I told, I'll tell you something about the glyoxysomes. Okay, glyoxysome is a kind of organelle. Okay, so it directly involves in the synthesis of carbohydrates from the fat. Okay, so as we all know, from the fat, okay, it will enters into the cycle called glyoxylate pathway. Glyoxylate pathway. Okay, so just remember one thing, glyoxylate pathway is the alternative pathway for the Krebs cycle. So Krebs cycle, from the Krebs cycle normally it will enters into the electron transport chain, but over here what happens over here, from the fat with the help of glyoxylate pathway, okay, it will synthesize the carbohydrates. Okay, so what are the major needs for the particular glyoxylate pathway? There are some specific enzymes need over here. Okay, so that is what mentioned over here. Some enzymes associated with the conversion of fats into carbohydrates. So what may be the role of the particular enzyme? They are going to involve in the beta oxidation. You have to remember the term beta oxidation. So what is beta oxidation? Beta oxidation is the enzymes, okay, with the help of enzymes, the fats are converted into carbohydrates. So where it happens? It happens in the pathway, glyoxylate pathway. What is the site of the particular glyoxylate pathway? The organelles, glyoxysomes. Okay, so you have to remember the term glyoxysomes, which contains the enzymes for the glyoxylate pathway. What is the role of the glyoxylate pathway? It is going to do the beta oxidation of fatty acids or fats into the carbohydrates. Okay, and see the next question. Which one of the protein is most abundant in the animal world? Which protein is most abundant or enormous in the animal world? So there are 4 choices for you. So the easiest choice you will pick insulin and hemoglobin. We all know our body will have a circulatory system, and the circulatory system will have a RBCs, and RBC is the matured RBCs is filled with a protein called hemoglobin. So we may think hemoglobin as a abundant protein in a human body or in any animal body, but the answer is wrong. We may think insulin as a abundant protein. That is also wrong, because insulin, as I told you, insulin involved in the metabolism of carbohydrates, especially the sugars. Okay, so if it is in a, it should be in a particular quantity. There must be some specific quantity should be available for a particular animal being. If it is more than a required amount, that is called hyperinsulin. Okay, that will cause a disease to the particular animal being. So you cannot choose the answer insulin. Insulin cannot be abundant in a particular animal. Well, insulin required. Insulin is a must protein for animal, but it must be in a specific quantity. It must be in a minimal quantity for the particular animal being. Okay. So see the next choices: trypsin and collagen. As I told you, trypsin is a kind of the protease. It is going to involve in the digestion of proteins. Trypsin is present in our digestive system. So what is the role of trypsin? It is going to involved in the digestion of proteins. It is going to degrade the particular enzymes. Trypsin is a protein molecule or enzyme molecule. What is the action of trypsin? It is going to act on proteins, and it is going to digest the proteins into polypeptides or into some other monomers of particular polypeptides. Okay, so you may think trypsin as an answer, but that answer is also wrong, because trypsin also available in a very minute quantity in our digestive system. So the answer over here you have to choose the collagen. So the collagen is an enzyme, collagen is a protein present in a muscle cells and all the fibers. Okay, so they are the building blocks of most of the tissues. So the collagen is the most abundant protein present in a animal world. See the final question. The enzymes are not found in. Okay, see the choices: algae, fungi, cyanobacteria and viruses. And you may get confused with the terms cyanobacteria, fungi and algae. Just remember one thing: all the living cells, they possess the particular enzyme groups. Okay, all the living cells, they are having the structures called enzymes. If there is no enzyme, then we cannot call that particle or that structure as a living cell. Okay, so you can easily eliminate algae, fungi and cyanobacteria, because they all belongs to a normal living organisms. Algae, fungi, cyanobacteria, they all having the particular structures called enzymes. So the choice over here, viruses. Okay, so you have to recall the thing about the virus. So viruses are the particles. Okay, so they are normally in a inactive form, inactive particles. Okay, so when they will get activated, once they are enters into another tissue. Okay, in a living tissue only they can be act as a dividing particle. Okay, once it enters into a biological living tissue. Okay, but the particular enzyme, they may possess the genes. They may having the particular genes for the proteins or for the enzymes, but as a structure within a virus there will be no enzyme. That is what you have to remember. In a virus, there are no enzymes, but they can have the particular, they may have the particular genes for the synthesis of the viral enzymes. Okay. So when the synthesis will happen, once the virus will infect the particular living cells, within a living cell, by using the living cell's own protein synthetic mechanism, okay, this virus will synthesize the particular enzyme. Okay, with the help of enzyme, it will make the particular virus particles so that it will multiply within the particular living cell. Then it will lyse the particular living cell and come out and infect, or it will enter into the other infective cycle. So the point here to remember over here from the question, enzymes are not found in as a cell. Algae will having the particular enzyme, fungi will having, cyanobacteria will having, but as a structure, viruses, they will not have the particular enzymes, but it will synthesize the enzymes once it enters into the living tissues. Okay, and these are the questions you can expect from the particular enzyme topic. In the next class, we are going to see something about the questions we can expect from the carbohydrates and lipids.